Hello everybody, my name is Grant Harrison. It is June 11th, 2023, and I'm here to talk to you about the anatomy and enzymology of fat absorption. As food enters the mouth, becomes chewed up and swallowed, there are numerous ways in which the foods we eat are processed. However, each macromolecule, lipids being one of them, or fats, has its own specialized method in which they are digested and lead to energy production. In this brief presentation, we'll focus on lipids or fats and how they're absorbed in the gastrointestinal system, or the GI. So as lipids enter the GI through the mouth, they are processed very minimally until they reach the stomach, or even I should say, in the stomach they are processed minimally, and then there is an enzyme present, gastric lipase, that is produced by the chief cells. We can see them here inside of the gastric mucosa, in the gastric gland. We can see these different chief cells, which actually secrete gastric lipase as well as pepsinogen that's used for protein synthesis, but that's neither here nor there for the purpose of this lipid presentation. So that one reason why there's not very much digestion of lipids in the stomach is because of the pH. The ideal pH for lipid digestion is 7. In the stomach, it's very acidic at 2 generally. So now we're going to talk about the binding, breakdown, and coating of lipids into micelles. So once out of the stomach, fats are much more easily digested due to the emulsification or physical breakdown of larger triglyceride molecules into smaller droplet molecules in the small intestine. It's important to note that the first step is only a physical breakdown and not a chemically or enzymatic breakdown. So once in the small intestines, lipids come into contact with micelles of bile salts secreting from the liver and gallbladder. Micelles may also be formed from the triglycerides when they come into contact with these bile salts. Those bile salts arrive from the bile duct and into the duodenum. And once in the duodenum, this is of the small intestine now, triglycerides accompanied by bile salts in the chyme are able to be broken down further into free fatty acids and a monoglyceride, which I will show you in the next slide. So here we have on the left-hand side, the left-hand image, you have a micelle. It is composed of diglycerides, monoglycerides, phospholipids, cholesterol, free fatty acids in the middle. We have bile salts along the outside. And those are going to be important. We're going to need a different enzyme other than lipase to be able to expose the inside of this molecule, and so it can be broken down further. At this point, enzymatic fat digestion occurs by lipase and colipase secreted by the pancreas. So colipase actually acts to replace some of the bile salts and make the inside of the lipid micelle accessible to lipase. So that's how colipase works, is it kind of flips it inside out, so to speak, or grabs onto it and makes it so lipase can actually bind and get in there and break up all these particles, which on the right hand side you can see now you can just imagine these being free floating in the lumen of the GI at this point in time. Alright, so now we have all these free fatty acids, monoglycerides, some cholesterols, phospholipids, but what we really want to focus on here is the free fatty acids, the monoglyceride. They're actually able to diffuse at the brush border of the epithelial layer. They're able to diffuse into the cells of the small intestine, the epithelial cells. And then at this point in time as well, cholesterol is also transported at this juncture. So they're making it into the cell, and in the cell, see again we have a formation of how the free fatty acids and cholesterol and monoglycerides are actually at the brush border. They make it in. So within the epithelial cell, however, the free fatty acids, monoglycerides, they actually enter the smooth plasmic reticulum and turn back into triglycerides. Proteins that have also made it into the cell combine with the triglycerides and cholesterol to form a chylomicron. Once a chylomicron will enter into the Golgi apparatus, form a vesicle, and be transported into the interstitial fluid via exocytosis. And then from there, here we go. From there, on the other side of the epithelial cell and into the interstitial space, chylomicrons do not readily absorb into the capillaries. Instead, chylomicrons are transported from the epithelial cells into the lymphatic system through specialized cells called lacteals. The lymphatic vessels eventually form a larger vessel known as the thoracic duct, which connects to the bloodstream at the junction of the left subclavian vein and the left internal jugular vein in the neck. We can see here a nice drawing of how we just went through the duodenum, through the small intestine, and now we're about to exit the small intestine and be absorbed, like I said, by the lymphatic system. And on the right hand side, so that will go up the trunk and enter into this location right here right between the left subclavian vein and the left internal jugular vein, as presented. So that is how your body processes and absorbs lipids or triglycerides, phospholipids, etc. Into the body. And these are my references, and thank you very much.